right, let's get started with advanced pathophysiology. Pathophysiology is basically the study of disease. So there's the breakdown of the word. Pathos and ology is the study of suffering, and physio is function, structure of organs. Put it all together, we basically get the study of disease. Pathophysiology are the changes that accompany a particular disease or syndrome. We are going to be looking at disease starting at the cellular level. We get alterations in molecules, genes, proteins, metabolites. All of that influences cell behavior and ultimately cell survival. All cells maintain cellular housekeeping, so they all have their certain roles. They all need to be able to function. They are all differentiated, and they must be able to perform their key responsibilities. If they're unable to perform them, then we have a breakdown, and ultimately we end up with cellular injury and cellular death. They maintain cellular viability by doing their jobs appropriately. We have isolations and compartmentalization, which is protective. As we go through cellular biology tonight, we're going to be looking at each organelle and how it is supposed to be behaving within the cell, and then when we see a breakdown in those organelles, then we get disease, and we'll talk about that moving forward throughout the whole course. The cells are protected from the environment. They have nutrient acquisition, they have metabolism, movement. They are unique, amazing little cells that do so much. So it's really awe-inspiring when we break it down and see exactly their responsibilities. We have to have normal functioning. We have to maintain intracellular homeostasis. If there's not intracellular homeostasis, then we ultimately have a breakdown and results in disease. A couple key terms of pathophysiology that you should understand. So pathogenesis is a little bit different than pathophysiology. So pathophysiology is understanding that entire kind of process. Pathogenesis is basically development of the disease. All right, the other way I like to look at it is the pathway. And we really look at, at that cellular level. The pathogenesis is how it goes from point A to point B, and we get the disease, okay? Etiology is basically the cause or origination of the disease. So this could possibly be genetics, environmental, autoimmune. Epidemiology is kind of who gets it, what age do they get it, where, is there certain geographical locations, is there specific populations? So we really break down, and this is from epidemiological data studies, and that is, we have a lot of statistics in this kind of category, epidemiology, and we'll talk about that as we move into more of the disease processes later on. Clinical presentation, so these are the classic signs and symptoms that accompany a disease process. Nosocomial is hospital acquired. Acute versus chronic, short onset, couple of days, couple hours, and then chronic are those more diseases that last for long term. Remissions and exacerbations. So remissions, the disease process may wax and wane. Exacerbations, you have a baseline disease process and something makes it worse. COPD is one of my go-to examples here. We get a pneumonia diagnosis and it makes the COPD worse. Sequela, this is a condition that is the result of another condition. Process that is the sequela of another disease process. And you have to code appropriately. Hip fracture, and then you get a fat embolism would be an example of that. So a condition that is the result of another condition. All right, idiopathic versus iatrogenic. Idiopathic basically means we really have no known cause. We don't know why this happens. We don't know the pathogenesis of it. If you see this in your studies, idiopathic, that's the answer, okay? There's nothing else for you to do. There's a lot of people that have looked into these disease processes and studied them, and when they come up with an idiopathic, it means that we just don't know why it happens. Iatrogenic is different. It means that healthcare causes is the best way that I can explain it to you. Whether this is, it's inadvertent most of the time. This is not malfeasant at all. Basically, you may do a diagnostic study, or you may prescribe a medication and something happens that causes another medical condition as a result of that. So a good example of this would be if I give you medicine to treat your hypothyroidism, and then you come back in with new signs and symptoms and you're actually hyperthyroid, that would be iatrogenic hyperthyroidism because it's actually related to the medication that I gave you. We're going to be looking at central concepts of pathophysiology. This really looks at cells and tissues, and then we're going to be looking at pathophysiological alterations in the actual disease organs and systems. We do have an entire lecture on genetics, so we just briefly going into this right now. We'll talk more about it in the genetics lecture. Remember that genes are our building codes composed of the sequences of DNA, deoxyribonucleic acid, located in the chromosomes in the nucleus, right? That's where we house all of this. We have 46 chromosomes, and they are 2 copies of 22 autosomes and 2 sex chromosomes. Just remember that it is phenotypically written like this, and it is basically our human genetic architectural design and the blueprint for what instructs everything that your body is going to do. We do have a lot of inherited genetic diseases due to the defects or mutations in the DNA. Ultimately, what we are getting with DNA transcription is protein synthesis, and anything that can result in mutations or disruption can affect protein structure, function, and disrupt that homeostasis. Remember that you have 4 nitrogenous bases, ACGT, and a double helix formation. Also, that genetic coding is nearly universal. Most organisms use the same DNA, and this is how common it is. So 98% human DNA is like chimps, 92 mice, 87 zebrafish, 84 dogs. This is why we do a lot of medication testing, unfortunately, because the DNA is so close to ours. We have 2 types of DNA. We have coding versus non-coding. There's about 3.2 billion base pairs. That's 98.5% of the human genome, and it does not encode for proteins. That is the non-coding DNA. It serves as gene expression, so it's basically the machinery that's required for cellular maintenance, provides the support, gives that scaffolding for the DNA that is coding to make those proteins and do what they need to do. The other 1.5%, it's about roughly 20 to 25,000 genes. Those are the protein coding genes, and that is what's going to get all of the proteins that are used within our body to do what's needed for our cell maintenance and making up DNA coding. DNA codes for proteins by amino acids, makes proteins. There is that classic picture of chromosome is this X-shaped structure. It's threaded very closely, supercoiled on top of each other. You can see the DNA is woven in between these little spherical proteins called histones, and we'll talk about those more in the genetics lecture. And then as the DNA is unwound, we get that double helix formation. And then here we have our nitrogenous bases that are paired. Remember, genes are basically a portion of the DNA, and they all have codes that they are given to them. About 80-85% of the genetic human genome has been coded for, so it's pretty cool. So this is the kind of gist of what happens with the DNA. And remember that this is in the nucleus, where the DNA is housed. Ultimately, cells divide. We need an extra copy of that DNA in the new cell, so there has to be an exact replica. And this is a very in-depth process. We'll go into it more in the genetics, but just want you to understand basically that we get the DNA, it's broken apart, we get a complementary base pair. DNA must be able to replicate itself accurately during that cell division so that the code can be preserved in subsequent cell generations. So very important that that happens. DNA replication consists of breaking of weak hydrogen bonds between the bases, so these bases over here, and then the single strand of each base is unpaired, and that consists of adenine with thymine, guanine with cytosine, known as complementary base pairing, so they match up. and that is the key to the accurate replication. Unpaired will attract a free nucleotide only if the nucleotide has proper complementary base. ATTGCT will bond with a series of free nucleotides with the bases of TAACGA, and that is hallmark of that complementary base pairing. Basically, it's a template or a guide, and the molecule on which the complementary molecule is built is the basis for synthesizing that new double strand. There are a few proteins involved in DNA replication. One protein unwinds the double helix, the other one holds the strand apart, and others perform distinct functions while we are doing the complementary base pairing. One of the most important ones is the DNA polymerase, and this enzyme travels down the DNA strand, adding correct nucleotides to the free end of each new strand. It also is a proofreader to make sure that there's no errors, and if there are errors, then it will actually correct them. So very cool. And it's on the next slide, so I do have all of this kind of written out for you. We do get mutations in that kind of area, and that is where we get base pair substitutions, missense, nonsense, frame shift, and we'll talk about that more. But these are where mutations can happen. Transcription is the process by which the RNA is synthesized from the DNA template. This result is the formation of the messenger RNA from the base sequence specified by the DNA molecule then. Transcription begins with an enzyme called RNA polymerase, and that binds to a promoter region, and that promoter region is a site of sequence of the DNA that specifies the beginning of the gene. In addition to the RNA polymerase, proteins called transcription factors, and then they have transcription factor binding sites for the timing of that transcription, on and off area, and that tells the messenger RNA exactly which genes to be coding for. Translation is the process by which the RNA directs the synthesis of the polypeptide, which is the protein. Messenger RNA cannot code directly for the amino acids. Instead, it works with tRNA, which is transfer RNA, and that is a strand of 80 nucleotides, and that is the site of the attachment of the amino acid. So again, this is very in-depth. I'm just kind of highlighting it right now. And then ultimately, we get the proteins. Every amino acid is coded by 3 consecutive bases to earn a codon. There are 20 possible amino acids and only 4 bases. Each single nucleotide cannot specify an amino acid. Similarly, there are amino acids that cannot be specified by a couplet of bases. Because there are only 4 by 4 or 16 possible couplets, there's a series of 3 bases are translated into amino acids, then there are 4 by 4 by 4, which equals the 64 possible combinations. And then we have 3 stop codons, which are the UAA, UGA, and UAG. So of those 3, we have 61 left, and those are protein coding. There is that classic picture of the cell. So all body functions depend on the integrity of those cells. How messages originate, how they're transmitted, received, interpreted, and used by the cell is hallmark. Cellular biology depends on that cellular communication. It is basically a streamlined conversation between, among, and within the cells that maintain cellular function and specialization. Organs and cells need homeostasis, right? So that's the end all be all. If we don't have homeostasis, then we're going to have cellular injury, cellular death, and ultimately disease processes. There will be changes, stressors. Any increase, decrease, or change in stressors can cause adaptations of cellular growth. If those adaptations are unable to be corrected, then we end up with cellular injury and cellular death. There are intracellular signals that determine the cell's position, function, and specialization, and are hallmark. This is called differentiation, and it is necessary for the cells to be able to maintain their tasks and then maintain homeostasis. We have 2 different types of cells. We have prokaryotic cells and eukaryotic cells. Prokaryotic cells are bacteria and archaea. Eukaryotic cells are plants, fungi, animals, and us, obviously. So prokaryotics have no nucleus. Their genetic material is not housed in a nucleus. You can see here it's more kind of free-floating. They also do not have any organelles, and they're characterized by that no nucleus. So I always remembered it, no nucleus. Eukaryotes, they're larger. They have a lot more intracellular organelles. They have a plasma membrane. They have cytosol, a nucleus that houses the genetic material. Let's take a look at the 8 chief functions of the cell. So cells become specialized through the process of differentiation or maturation, and some cells eventually perform one kind of function, other cells perform another kind of function. So not all of these cells have kind of all of these functions together. It all depends what type of cell it is. Conductivity is a response to the stimulus that's manifested in a wave excitation. So we have stimulus, and then we get excitation. So think your electrical stimulus that happens in your neurons, or the chemical electrical stimulus that happens in muscle cells. So there is a conduction as a response to stimulus, wave of excitation, electrical potential that passes. Metabolic absorption, so all cells take in nutrients to carry absorption. So that would become some of the specialized cells here. So stomach, kidney cells, and then conductivity, so think muscle. Secretion, certain cells have the ability to secrete other substances, so think mucus, glandular cells, and they can secrete a secondary substance. Movement, muscle cells generate force. Muscles are attached to the bones, they produce limb movements. There's hollow tubes or cavities and empty contents basically when they contract, and we'll go a lot more into this when we get into the muscle cells. And then we have surrounding blood vessels, change in diameter. There's a whole wave of movement that happens as we generate forces that produce motion. Excretion, all cells must be able to rid themselves of waste products resulting from metabolic breakdown of nutrients. We have lysosomes, which we're going to talk about in a few minutes, that enzymes that break down, digest large molecules, and then turns them into waste products that are released from the cell. Respiration, so cells absorb oxygen, and then that is utilized to make ATP, which is energy that all our cells utilize. That's adenosine triphosphate. Cellular respiration or oxidation occurs in the mitochondria, and we're going to go over that later. Reproduction, tissue growth occurs, cells enlarge, reproduce themselves. So we basically have reproduction of cells, regeneration, and tissue maintenance requires new cells be produced or replace the cells that are lost normally, in cellular death. We'll talk about cell senescence a little bit later as well. Communication is vital for all cells to be able to maintain their viability. They need to be able to communicate back and forth, allow for that steady state and homeostasis. We're going to go into each and every one of these. Again, there is that classic picture of the cell. This is that typical eukaryotic cell. Has 3 main components. That is the outer membrane called the plasma membrane, fluid filling called cytoplasm, and then we have all these intracellular organelles. First up is the nucleus. So this is surrounded by the cytoplasm, generally located in the center of the cell, largest membrane-bound organelle. We have this nuclear envelope, and that is pockmarked with these pits called nuclear pores. Also part of these nuclear pore complexes, and they allow molecules to move between the nucleus and the cytosol. The outer membrane is actually attached to the endoplasmic reticulum. This is where the genetic information is housed. Primary function is cell division and control of that genetic information. Replication, repair of DNA, transcription of the information stored in the DNA, and transcribed into RNA, which is processed into messenger transport and ribosomal RNA, which we talked about a little bit earlier.